Hello, everyone, and welcome. Today, we are going to explore a fantastic topic, biotechnology. But I want us to think about it in a slightly different way. Let's forget the difficult textbook chapter for a moment, and instead, let's imagine it as a fascinating recipe, a recipe for life itself.
So let's start with a question that honestly seems like it's straight out of a science fiction movie, no? How on earth can we ask a simple, tiny bacterium to produce something as complex as human insulin for us? Well, this is the very puzzle that biotechnology helps us solve, and I promise you, by the end of this explainer, you will understand exactly how it's done.
So to answer that big question about insulin, we first need to understand the big idea behind all of this. What is biotechnology, really?
In very simple words, you can think of it like this. We are basically becoming chefs, but on a microscopic level. Instead of using normal food ingredients, our ingredients are living things, like microbes, plants, or even animal cells. And we use these living ingredients to create products that are super useful to us, like life-saving medicines and vaccines.
Now, you might be surprised to know that this idea isn't new at all. We've been doing this for centuries. Making curd from milk, or baking bread, or making wine. That is all traditional biotechnology. We're using a whole organism, like bacteria or yeast. But modern biotechnology, which is our focus today, is much, much more precise. We're now focusing on the very code of life itself, the DNA.
Modern biotechnology really stands on two strong pillars. The first one is genetic engineering. This is the master technique, where we can actually change the chemistry of DNA and RNA. It's like rewriting the recipe book itself. And the second pillar is bioprocess engineering. This is all about creating the perfect clean kitchen, a sterile, contamination-free environment for our tiny engineered organism to grow and make the product for us. Both are absolutely crucial.
Okay, so every good recipe needs the right tools and ingredients, right? In genetic engineering, we have a very special molecular toolkit. So come on, let's open it up and see what's inside.
First up, we have our molecular scissors. These are special proteins called restriction enzymes, and they are amazing. They can read a long DNA strand and make a precise cut only at a very specific recognizable sequence. Now for your exams, you must remember that these sequences are typically palindromic. That means they read the same on both strands when you read in the 5' to 3' direction. A very famous example is EcoRI, which always recognizes and cuts the sequence GAATC.
And this brings us to a very important point, especially for entrance exams. These molecular scissors can cut in two main ways. Some, like our friend EcoRI, cut the DNA strands a little away from the center of the palindrome. This leaves short, single-stranded overhangs that we call sticky ends because they can easily pair up with a complementary end. Other enzymes cut straight down the middle, making blunt ends. Now for our recipe, sticky ends are much, much easier to work with. Remember this difference. It's a very common MCQ topic.
So, if you have scissors to cut, you must also have some glue to join things back together, isn't it? Well, our molecular glue is an enzyme called DNA ligase. Its only job is to join two pieces of DNA together by forming strong bonds, permanently sticking them together.
Now, a very important question. How do we get our desired gene into a new cell? For this, we need a delivery vehicle, or what we call a vector. Think of it like a tiny truck that carries our precious genes safely inside the host. A very common example of a vector is a plasmid, which is a small circular piece of DNA found in bacteria.
And finally, we need a factory. This is our host organism, where our gene will be copied and our product will be made. For this, we usually choose a simple and fast-growing bacterium like Escherichia coli or E. coli for short. This little guy is truly the workhorse of biotechnology.
So we have our tools all laid out. We have our scissors, our glue, our delivery vehicle, and our factory. Now let's get to the main method. How do we put all of this together to create something called recombinant DNA?
The entire process can be broken down into these 4 simple and logical steps. Let's look at each one slowly and carefully.
Okay, the first 2 steps are isolation and cutting. So we take out the human DNA, which has our insulin gene, and we also take out the plasmid vector DNA from the bacteria. Now, here is the crucial point, and please pay close attention because this is a very common exam question. We cut both the human DNA and the plasmid DNA with the exact same restriction enzyme. Why? To create matching, complementary sticky ends that can join together perfectly.
Now comes the magic part.
We simply mix our cut gene and our cut plasmids together in a test tube. Because they have complementary sticky ends, they will naturally find each other and pair up. Then, we add our molecular glue, DNA ligase, which seals them up permanently. And just like that, we have created our masterpiece, a new hybrid DNA molecule, which we call recombinant DNA, or rDNA for short.
The final step in our recipe is called transformation. We need to introduce this new rDNA into our host bacterium factory, like E. coli. But you know, a bacterial cell won't just take up foreign DNA easily. We have to make it competent to do so. We usually do this by treating it with a specific concentration of a divalent cation, like calcium, and then giving it a brief heat shock. This helps the rDNA to enter the bacterial cell.
But hang on. What if we only have a tiny amount of our gene to begin with? Before we make the final product, we need many copies of our gene. For this, we use a magical machine and a really powerful technique.
This technique is called PCR, which stands for polymerase chain reaction. The best way to think of it is as a molecular photocopier. It's an incredible method that can take just a single copy of a DNA segment and amplify it, making billions of copies in a very short time.
The PCR cycle has 3 main steps that are based on changing the temperature, and these steps are repeated again and again. First is denaturation. We heat the DNA to a high temperature, around 95 degrees Celsius, to separate the 2 strands. Second is annealing. We cool the temperature down, which allows small pieces of DNA called primers to bind to our target sequence. And third is extension. We raise the temperature to about 72 degrees, and a special heat-stable enzyme called Toc polymerase starts adding new bases and builds the new DNA strand. This Toc polymerase is a hero, really. It comes from a bacterium called Thermus aquaticus that lives in hot springs, so it doesn't get destroyed at the high temperatures. We just repeat this cycle 30 or 40 times, and we get billions of copies.
Okay, we have our engineered bacteria, and they are ready to work for us. But how do we get our final product, like insulin, in large enough quantities to help millions of people? We need to scale up.
The answer is a bioreactor. And this is not a small test tube in a lab, okay? A bioreactor is a massive, controlled container where our bacteria can grow and produce our protein on a huge industrial scale.
And when I say massive, I really mean it. Bioreactors can be huge, holding hundreds to thousands of liters of culture. Just imagine thousands of liters filled with billions and billions of tiny bacterial factories, all working to make our desired protein.
Now, these are not just simple tanks. They are highly complex and sophisticated machines. Inside a bioreactor, there are systems to control everything perfectly. There's an agitator system for constant mixing, an oxygen delivery system, a foam control system, and sensors to check the temperature and pH at all times. It's all about creating the perfect conditions to keep our little factories happy and super productive. You know, we started with a question: how can a bacterium produce human insulin? And now you have the answer. You understand the complete recipe for biotechnology. So I will leave you with a final thought. Now that you know this incredible, powerful recipe, what revolutionary product would you create to change the world?